हॅलो, एम आय ऑडिबल? हॅलो, आवाज येतोय का? हॅलो, आवाज येतोय का? येस. ऋतू शेलार, आवाज येतोय का? प्राजक्ता मोरे, आवाज येतोय का माझा? हॅलो, माझा आवाज येतोय का?
हो सर, येतोय आवाज.
आणि पीपीटी दिसते का?
नाही.
पीपीटी दिसत नाही? आता बघा की. हॅलो, पीपीटी दिसते का?
नाही.
हॅलो, कोण? बर, बोला. आता मॅडम मी लेक्चर मध्ये नंतर करा लेक्चर मध्ये. पीपीटी दिसत नाहीये. आता दिसते का पीपीटी? हॅलो,
नाही सर,
नाही.
नाही दिसत.
आता दिसते का रे पीपीटी बाळांनो? हॅलो, प्रोजेक्टचा पीपीटी दिसते का आता?
नाही दिसत सर.
नाही दिसत
नाही
सर.
दिसत आहे.
नाही.
मग काय झालं रे? आता दिसते का रे?
हो सर.
दिसते.
दिसते ना? ओके ओके ओके. Okay, today we would like to finish this methods of food preservation. Of these methods of food preservation, methods for you people, that is asepsis technique, that is keeping microorganisms away from the food material and keeping microorganisms away from the food material. Various techniques are there. Asepsis technique are there. So in near future, earlier pen and paper technique is there. If it is there in future for examination, this asepsis and removal of microorganism, these two techniques are important. These two techniques are important, asepsis and removal. Then we have seen this last time. We have seen this also seen. Okay. Asepsis we have seen in. And next to asepsis, one of the most reliable and promising technique last time we have seen, that is the use of a high temperature. And in case of use of high temperature, basically in case of food, in your syllabus they have been mentioned pasteurization and sterilization. This pasteurization is use of low temperature for long time. That is temperature used in case of pasteurization is 62.8 degrees centigrade for half an hour, and this is basically used for what you can say this milk pasteurization. And the index organism of pasteurization are Coxella burnetii and Mycobacterium tuberculosis. Amongst these pathogenic organisms, these two organisms are the most heat-resistant microorganisms, and therefore these organisms gets killed below the temperature used for pasteurization. Below the temperature used for pasteurization. Then the high temperature, that is time temperature cycle you have to use, and one of the most reliable and promising high temperature, that is 121 degrees centigrade for 15 to 20 minutes is the reliable temperature. But at 121.6 degrees centigrade for half an hour, depending upon nature of the food material, the carbohydrate food material is there, then caramelization may take place. Therefore, depending upon the nature of the food material, you have to design or decide time and temperature cycle, and for that purpose also you have to take into the consideration various terminologies, that is TDT, TDP, then D value, F value, Z value. Last time we have seen all these. Then you have to take into the consideration at a temperature of incubation, phase of growth, whether it is lag phase, log phase, stationary phase, whether presence or absence of hydrogen ion concentration. All these points you have to take into the consideration. And last time we have discussed about thermoduric, thermophilic, and one more point, that is by use of temperature. Last time I might have been told you people, that is aseptic packaging. In aseptic packaging, that is a traditional canning technique is called. And in this traditional canning technique, non-sterile food is placed in non-sterile metal or glass containers and followed container closure and sterilization. This is one technique used for food preservation. That is non-sterile food is placed in a non-sterile cans, and then it is followed by sterilization. And one more, aseptic packaging, that is sterile food under aseptic condition is placed in sterile containers. This is also one technique for food preservation. Then next, preservation by use of low temperature. So as all of you are aware, that is the vital metabolic activities are carried out by the enzymes, and enzymes, as all of you, require a specific temperature or certain temperature. So what happens at low temperature? At low temperature, vital metabolic activities or enzyme activities are decrease. So here they have been mentioned. Low temperatures are used to retard chemical reactions and action of food enzymes, and to slow down or to stop activity of microorganisms in food. The lower the temperature, the slower will be the chemical reactions as well as enzyme action. And microbial growth, and low enough temperature will prevent the growth of any microorganism. So any raw plant or animal food may be assumed to be contain variety of bacteria, yeast, molds, algae, fungi, which needs to only conditions for growth to bring about undesirable changes in the food. And by application of low temperature will prevent the growth of these desirable organisms, but slow metabolic activity may continue. The growth and metabolic reactions of microorganisms may depend upon the enzymes, and the rate of enzyme reactions are directly affected by the temperature. So here I would like to tell you people, preservation by use of low temperature. As all of you are aware, in our day-to-day life, we are preserving the prepared food by keeping in freeze at lower shelf, where temperature is up to 10 degrees centigrade, or from 0 to 10 degrees centigrade, whereas in upper shelf, where we call freezer, the temperature is 0 degree centigrade or below 0 degree centigrade to some extent. So what happens, I would like to tell you people that preservation by use of low temperature. So you have to take into the consideration what is the preservation temperature. What is the preservation temperature? If preservation temperature is sufficient to stop entire metabolic activities of microorganism, then we can preserve the food for considerable period of time. But sometimes what happens? Only 49 are there. Only 49 are there. Is इथे प्रिझर्वेशन बाय लो 48च आहेत का? आज सकाळी झालं नाही तुमचं लेक्चर कुठलं? येस, सकाळी कुठलं लेक्चर झालं नाही? हा, माझंच पहिलं आहे. पहिल्या लेक्चरलाच दांडी एवढ्या जणांची. अरे बाप, कसं रे बाळांनो कळणार तुम्हाला? मला काय सांगायचं होतं, प्रिझर्वेशन बाय यूज ऑफ लो टेम्परेचर मध्ये जे काही टेम्परेचर आपण वापरणार आहे त्याच्यावरती आपलं प्रिझर्वेशन पिरियड पुढचा ठरणार आहे. दॅट यू हॅव टू कीप इन युअर माइंड. सो ग्रोथ ऑफ मायक्रोऑर्गॅनिझम ऑकर ऍट लो टेम्परेचर. So it has been given some of the example. For example, some of the moods, cladosporium, sporotrichum. It has been found to some of the scientists at minus 6.7 degrees centigrade, whereas penicillin growth, it was found at minus 4.0 degrees centigrade. And some of the yeast they have found even at minus 34 degrees centigrade. And bacteria have been reported growing at temperature as low as minus 5 on meats, and minus 10 on cured meats, and minus 11 on fish, and minus 12.2 on some of the vegetables. That point also you have to take into the consideration. So temperature employed in low temperature storage is usually it is called as a frozen storage. Many of the terms used in connection with the low temperature For example, frozen storage, one term is used, and the temperature of frozen storage, that is deep freezes, it is below minus 18.0 degrees centigrade. Below minus 18.0 degrees centigrade. This is also called as a cold storage preservation. This is also called as a frozen storage. This is also called as common or cellar storage, that is below 15 degrees centigrade. Root crops, potatoes, cabbage, celery, apples, similar foods can be stored at this temperature. One more term is coined to this temperature, use of low temperature, that is cold storage or chilling, is at temperature not far above freezing and usually involves cooling by ice or mechanical refrigeration. It may be used as the main method of food preservation for temporary preservation. Most perishable foods, for example, eggs are there, dairy products are there, meat products are there, or meat, fresh meat is there, seafood is there, vegetables and fruits are preserved by this. Again, just now I have told you people freezing or frozen storage. The storage of foods in the frozen condition has been an important preservative method since long time. With the development of mechanical refrigeration and the quick freezing process, the frozen food industry has nowadays expanded largely, or it is nowadays still expanding rapidly. Even at home, the freezing of food has become extensive now that home deep freezers are easily available. Freezing of foods, the rate of freezing of foods depends upon number of factors such as method employed, the temperature, circulation of air, size and shape of the package, kind of food, etc. That also you have to take into the consideration while carrying out the deep freezing of the food. Then sharp freezing usually refers as a freezing in air with only air circulation or best electrical fan, and the temperature usually used is minus 23.3 degrees centigrade or lower, but it varies from 15 to 29 degrees centigrade. And freezing may take from 3 to 72 hours. Sometimes this is referred as a slow freezing. In contrast to slow freezing, the quick freezing is accomplished by direct immersion of food or packaged food in a refrigerant. As in the refrigerating of a fish in brine, the temperature of brine is usually minus 30, 40 degrees centigrade. Secondly, by the indirect contact with the refrigerant coolant agents are there, some of these chemical having minus temperature below 30, 40 degrees centigrade. And thirdly, by air blast freezing, where frigid air at minus 17 to 35 degrees centigrade is blown across the material to be frozen. Then so there is a chance if what you can say, this earlier method of examination is there, preservation of food by low temperature. Under that circumstances, you will write preservation by low temperature. Temperature use the technique of preservation by low temperature. So anybody is having a doubt or difficulty in case of low temperature. Last time we have seen the high temperature. लो टेम्परेचर लिखता येईल का 4 मार्क्सला शॉर्ट नोट लो टेम्परेचर मेथड ऑफ फूड प्रिझर्वेशन हाय टेम्परेचर लो
सर
हाँ येस
13 नंबरची स्लाइड परत दाखवता का जरा
13 नंबरची स्लाइड परत दाखव का yes this is related to high temperature This is related to high temperature. You have to take all these points into consideration when you are carrying out preservation of food by high temperature. That is temperature of incubation after application of the high temperature. Phase of growth, कुठल्या फेज मध्ये ऑर्गॅनिझम्स आहेत, लॅग फेज मध्ये आहेत, लॉग फेज मध्ये आहे, कोणत्या प्रकारची हीट वापरलेली आहे, moist heat वापरलेले आहे, dry heat वापरलेले आहे. Moist heat is having high penetration power, dry heat is having low penetration power, then pH of the food. हे सगळे कन्सिडर करायचे असतात पॉइंट. Who is asking me regarding the 13 number slide?
सर मी संयुक्ता.
Yes, Sanyukta, please
समजलं
ask
सर.
me. Sanyukta, please ask me what is your doubt or difficulty regarding the 13 number slide. I feel मला असं तुमच्याकडून सुद्धा टू वे कम्युनिकेशन असेल तरच मला जरा शिकवायला इंटरेस्ट येत असतोय. अदरवाईज काय मी एका बाजूने सांगत जातोय, तुम्ही अजिबात रिस्पॉन्स काय देत नाही. म्हणजे काय की मला कुठेतरी याच्यात अनोळख्या ठिकाणी काहीतरी चालल्यासारखं वगैरे बऱ्याचशाच वेळेला वाटत असते. तर संयुक्ता, what is your problem in slide number 13? So all of you can
सर
ask.
ते मिस झालं होतं जरा माझं.
Miss झालं होते. I thought you have
त्यामुळं
got doubt in slide number
नाही,
13.
इझी सर.
Apart from Sanyukta, anybody is having doubt or difficulty regarding the technique we are discussing about the food preservation technique वरती 110% प्रश्न असेल. जर पूर्वीचीच एक्झाम टेक्निक असेल तर इट इज रादर डिफिकल्ट टू प्रेडिक्ट इट नाऊ. इट विल बी अर्ली. कारण अजून आपल्याला जे काय COVID-19चं जरी दिवाळीनंतर जरी सांगितलं असेल तरी सुद्धा काय अजून परीक्षेबद्दल आपण जे टीचिंग चालू आहे त्याच्याबद्दल कोणतीही गव्हर्नमेंट कडून विद्यापीठाकडून तशा प्रकारच्या गाईडलाईन्स आलेल्या नाहीत. वेदर युअर एक्झामिनेशन विल बी एंटायर MCQ क्वेश्चन्स वगैरे. तर थोडस आपणाला त्या दृष्टीने पाहावं लागेल की आपलं कॉलेज काय ते 50% वगैरे चालू झाल्यानंतर. तर काय शंका असेल तर बाळांनो विचारायची आहे. अदरवाइज वुड लाईक टू स्विच ऑन फॉर another most important technique, that is what you can say this preservation by drying. Preservation by drying. Yes, ऐकलाय का शब्द preservation by drying? Yes, anybody from class?
येस सर.
हा, तर प्रिझर्वेशन ऑफ ड्राईंग मध्ये सगळ्यात महत्वाचं लक्षात ठेवायचं काय, ग्रेन सगळ्या प्रकारचे ड्राय केले जात असतात किंवा सगळ्या प्रकारचे ड्राय फ्रुट्स आपण जे खात असतो ते केलेले असतात किंवा आपण ज्याला रेझिन्स बेदाणे असं म्हणत असतो त्याचंही ड्रायिंग केलं जात असतं किंवा आपणा सर्वांना माहिती आहे की मिल्क मध्ये 85% पाणी असतं. 85% water is there in the milk, but by removal of water from the milk, drying of the milk is carried out. So preservation by this drying is usually accomplished by this removal of water. So moisture may be removed from the foods by many methods. Sun drying technique is there, mechanical dryers are there, freeze dryers are there, drum drying technique is there, drying during smoking is there. So preservation by this drying, and one of the most important factor we have seen, that is factors that is what you can see this that is the intrinsic factor and the natural factor of the food material, that is available water or water activity. There is a relationship between water activity, available water which is required for vital metabolic activities, and drying. So the basic principle involved in drying, that is the removal of water by the natural way or removal of the water by use of certain specific techniques. And as a result of removal of water, water is no more available for vital enzymatic activities or metabolic activities, and as a result there is no growth of microorganisms. As a result there is no growth of microorganism. So what happens? Preservation by drying. Will you able to write preservation by drying? Yes, प्राजक्ता मोरे 2 आहेत. कसं काय रे बाळांनो? प्राजक्ता मोरे 2 Prajakta More. Yes. Lithail Kabalanu, preservation by drying, method of food preservation. Siddhi Shinde. Siddhi Shinde. Lithail का नक्की त्याच्यानंतर प्रिझर्वेशन बाय फूड ऍडिटिव्ह ऐकलाय का शब्द प्रिझर्वेशन बाय फूड ऍडिटिव्ह फूड ऍडिटिव्ह ऐकलाय का शब्द येस हॅव यू हर्ड वर्ड फूड ऍडिटिव्ह बळा ऐकलाय का शब्द प्रिझर्वेशन बाय
येस
फूड
सर प्रिझर्वेटिव्ह सारखं
प्रिझर्वेटिव्ह So preservatives are basically chemical substances or the natural preservatives. We call it as organic preservatives. Some of the preservatives are produced by the microorganism. For example, all of you are aware the organic acids which are lactic acid, acetic acid, or other propionic acid. These acids are produced by the microorganism. To some extent, these organic preservatives are also used by the microorganisms. By used as a preservative. So, preservation by food additives. A food additive is a substance or a mixture of a substance other than the basic foodstuff, which is present in food as a result of any aspect of production, processing, storage, or packaging. The term does not include chance contamination of the additives, according to who. This is an intentional additive पर्पजली ऍडिशन केलेल्या असणाऱ्या केमिकल सबस्टन्सला फूड ऍडिटिव्ह असं म्हणत असतात अँड बाय ऍडिशन ऑफ फूड ऍडिटिव्ह स्पेशली फूड ऍडिटिव्ह टू ऍडेड टू प्रिव्हेंट deterioration or decomposition of the food. And the food additives, just now I have told you people, they are of basically two types: chemical food additives and biological food additives. As far as chemical food additives, according to your reference literature, benzoic acid and parabens are there, sorbic acid and their substrate compounds are there, the propionates are there, sulfur dioxide and sulfides are there, nitrites and nitrates are there, NaCl and sugars are there, indirect antimicrobials are there. Indirect antimicrobials, n number of the antibiotics are also used for preservation of food. Acetic and lactic acid, just now I have told you people. Antibiotics are there and miscellaneous chemical techniques are there. Miscellaneous chemical techniques are there. Chemicals are considered in case of chemicals. Today, one table is there on page number 261. That is food microbiology by modern food microbiology by James and MJ. And in that today, it is rather difficult to show you people. According to that table, name of the preservative is there. For example, propionic acid and propionates. Propionic acid and propionates are there. The maximum tolerance or maximum concentration is 0.32% propionates and propionic acid. The organism affected by these propionates and propionic acids are mainly molds. Then the food which is preserved by addition of propionic acid and propionates are bread, cake, some cheeses. Then it is used to inhibit rope inhibitor in bread duff. That is a formation of rope in bread duff. For that purpose, propionates and propionic acid, and mostly it is used for inhibition of molds, and maximum concentration used is 0.32%. Next to propionic, sorbic acid. Sorbic acid. So the concentration of sorbic acid is 0.2%. Concentration of sorbic acid is 2%. Again, it is used for to inhibit the growth of molds, and it is basically used for this sorbic acid is used for hard cheese, figs, syrups, salads, jellyfish, jellies, cakes, dressings, etc. type of the food. That is sorbic acid and sorbets. Sorbic acid and sorbets. Then next is the benzoic acid. Benzoic acid, the percentage used is 0.1%. 0.1%. And it is mainly used for yeast and molds. It is mainly used for yeast and molds inhibition. Then it is used for pickle, apple cider, soft drinks, tomato ketchup, salad dressings, etc. Then parabens. Parabens, again, concentration use is a 0.1%. 0.1%, and it is used for yeast and molds inhibition, and the food products preserved by these parabens are bakery products, soft drinks, pickles, salad dressings, etc. Next to that, the sulfur dioxide and sulfides. The concentration used is 200 to 300 ppm, that is parts per million, that is 200 to 300 milligram per liter. And it is used, the organisms affected are insects and microorganisms. And the sulfur dioxide, it is used for molasses, dry fruits, wine making, lemon juice, etc. And next to this sulfur dioxide and sulfides, some of these other, that is ethylene propylene oxides are there, and concentration of ethylene propylene oxide is 700 ppm. It is mainly used for yeast and molds, and it is used as a fumigant for species, for some spices as well as nuts. Then sodium acetate and dehydroacetic acid, sodium nitrate, capralic acid, ethylene formate, some of the examples have been mentioned. And in addition to that table, NaCl and sugars are also used for preservation. As all of you are aware, NaCl and sugar for effect of osmotic pressure. Effect of osmotic pressure. So what happens? So this antibiotics, niacin antibiotic is there. Then natamycin antibiotic is there. Tetracycline is there. Subtilin is there. So these antibiotics, tylosin antibiotic is there, and tylosin antibiotic is there. Yes. याच्यानंतर आता जो काय मी एक पॉइंट सांगितला, प्रिझर्वेशन ऑफ द फूड बाय यूज ऑफ ऍडिटिव्ह केमिकल प्रिझर्वेटिव्ह. सो बऱ्याचशाच वेळेला आपल्याला असं सांगितलं जात असते की केमिकल प्रिझर्वेटिव्ह वापरलेले फूड खाऊ नये, बिकॉज व्हॉट हॅपन्स, ऑल दीज केमिकल प्रिझर्वेटिव्ह आर carcinogenic in nature. They can cause these mutations, they can cause changes, and as a result of changes or mutation, what happens, they may cause cancer. तर याच्या हा जो काय आता जस्ट नाव मी सांगितलेला पॉइंट कोणता, प्रिझर्वेशन ऑफ फूड बाय फूड ऍडिटिव्ह, फूड प्रिझर्वेटिव्ह, and in that food preservatives, biological food preservatives as compared to chemical food preservatives are good for health. आता थोडक्यात मी जे काही चार्ट सांगितला तेवढा चार्ट लक्षात ठेवणं गरजेचं आहे. अदरवाइज व्हॉट विल हॅपन when you go through this food microbiology by J and James, about 100 pages information is there. More than 100 pages information is there for all the food techniques. So in short we have to see the basic principle of the food preservation in short. So all these food preservatives in detail, that is their mechanism, it is not expected. So next year we may change the syllabus and of these various methods we can incorporate only one method in detail, but this year it is not mentioned in your syllabus. The methods of food preservation, they have been mentioned, therefore it is necessary to have highlight on all these methods, that is keeping of the food asepsis, removal of the food, removal of the microorganisms, use of low temperature, high temperature, then use of chemical preservative, etc. Chemical preservative, and the another method of the food preservation, that is food preservation by irradiation. The last method of food preservation by irradiation. Irradiation. तर काय I would like to ask you people, हा शब्द ऐकलाय का, radiation or irradiation? Radiation or irradiation word, have you heard this word? Irradiation, radiation. ऐकलाय का शब्द बाळांनो? Yes.
Yes, sir.
Sir, what is your perception about the radiation or irradiation?
Radiations are the rays produce म्हणजे आपण इलेक्ट्रोमॅग्नेटिक असे इन्स्ट्रुमेंट्स यूज करून रेज पास करायच्या फूड मधून.
Food madhun. Very good, very good. When we'll go through this scientific literature, we'll find that food preservation by irradiation. So in 1929, a patent was issued for use of radiation for food preservation in 1929. But during this World War First and after World War Second, the maximum effort was carried out for preservation of food by irradiation. So radiation may be defined as emission and propagation of energy through space. Radiation may be defined as emission and propagation of energy through space or through a material medium. This is called as a radiation. And these radiations have a very small radiations and a large radiations. The type of radiation of interest in food preservation are electromagnetic radiation. Apart from electromagnetic radiations, the what you call this, the radiations which are coming from the sunlight, that is ultra sunlight, that is what you call, that is the visible light, light, as well as this electromagnet radio waves, and what we call as a UV light. So all these radiations coming from the sun, and they have various wavelengths, and depending upon the wavelength, separation of these radiation is carried out. So here I would like to tell you people, the type of radiation of interest in food preservation is electromagnetic radiations. The various radiations are separated on the basis of its wavelength. Shorter the wavelength, the most damaging effect to the microorganism. Shorter the wavelength, having the high penetration power. The electromagnetic spectrum further divided into microwaves, ultraviolet, gamma rays, X-rays, etc. The radiations of primary interest of food preservation are ionizing radiations. Ionizing radiations may be defined as those radiations that have wavelength of, that is 2000 angstrom or 200 nanometer. 2000 angstrom or 200 nanometer or less. And the classic examples are alpha particles, beta particles, gamma rays, X-rays, and cosmic rays. The quanta contain enough energy actually to ionize the molecule in their path, since they destroy microorganisms without increasing the temperature. The process is called as a cold sterilization. Process is called as a cold sterilization. Process is called as cold sterilization. हा शब्द ऐकलाय का, cold sterilization? Have you heard this word, cold sterilization, anywhere? Yes, Prajakta? Yes, Prajakta? Siddhi Shinde? Tanvi Kale? What I am asking you, have you heard the word cold sterilization? Yes. अंजली कदम, रितू शेलार, शिल्पा दळवी. हाँ, what is your perception about cold sterilization? रितू शेलार, कोल्ड स्टरलायझेशन म्हणजे काय बाळ? रितू, येस, कोण सांगू शकेल? अबोली भिलारे, तन्वी काळे, अस्मिता डिके, येस. Control by eradication, that is in cold sterilization, that is sterilization by ionizing radiations where heat is not generated and ionization takes place so the ionizing radiations मध्ये काय मला पाहायचं आहे? You might be aware of the spectrum of what you can say the spectrum chart, that is electromagnetic spectrum. On the left side the cosmic rays are there, gamma rays are there, x-rays are there, ultraviolet rays are there, and then visible portion of the light where violet, blue, green, yellow, orange and a red light is there. That is what you can say this 400 to 800 nanometer wavelength. We call it as a visible spectrum of a light. Then below the visible spectrum of a light, what we call this UV spectrum of light. Below this visible spectrum, UV spectrum of light, where where the wavelength is in the 136 or 10 nanometer, 10 nanometer to 200 nanometer, 10 nanometer to 10 nanometer to 200 nanometer. या वेव्हलेंथला आपण अल्ट्राव्हायलेट वेव्हलेंथ म्हणत असतो. आणि या अल्ट्राव्हायलेट वेव्हलेंथ मध्ये सुद्धा आपण काय करत असतो? We are using some of these rays for eradication. So last year you might have been studied effect of ultraviolet light, that is they can cause this what you can see this the quantum in case of ultraviolet light, powerful bacterial agent with most effective wavelength at 260 nanometer, 260 nanometer. दोन मिनिटांनी 200 and ultraviolet light is a powerful bacterial agent with most effective wavelength about 260 nanometer. It is a non-ionizing and absorbed by the proteins and nucleic acids in which photochemical changes are produced that may lead to the cell death. The mechanism of UV, ultraviolet death in bacterial cell is due to the production of lethal mutation. Just now I have told you people as a result of action of upon cell nucleic acids. So तर काय लक्षात ठेवायचं? इरॅडिएशन्स मध्ये अल्ट्राव्हायलेट रेज लक्षात ठेवायचे. ते अल्ट्राव्हायलेट रेजची वेव्हलेंथ किती वापरायची आहे? 260 nanometer wavelength is one of the most effective wavelength, and at that wavelength it is a non-ionizing radiations absorbed by the proteins and nucleic acids present in the bacterial cells in which photochemical changes are produced, and that photochemical changes may lead to the cell death. And in case of bacteria, the death mechanism is due to the production of lethal mutations, and that lethal mutations, as all of you are aware, in natural what you can say this complementary strands, that is adenine always pairs with thymine, whereas cytosine pairs with guanine. So that pair that pairing changes in case of when we expose microbial cells to the ultraviolet light, and as a result of this pairing of that the mutation occurs, and as a result of mutations further growth, further what you can say this the uh, generation of DNA does not occur, and as a result death may occur. तर इथं काय लक्षात ठेवायचं आपल्याला, use of ultraviolet light for the preservation, use of ultraviolet light as a preservation. In a similar fashion, beta rays are also used, in gamma rays are also used, and these gamma rays are obtained from cobalt 60 and cesium 137. Those are the radioactive substances from that they are obtained, from them they are obtained. So what happens, what happens, food preservation by this radiation, food preservation by this radiation मध्ये काय करायचं आहे बाळांनो तुम्हाला, basically you have to take into the consideration a rad, the dose of radiations you have to take into the consideration, and depending upon the dose, whether that dose is a lethal to or it is inhibitory to enzymes, etc., that first you have to check it, and then that dose you have to use it. तर हे food preservation by irradiation, it is also a big technique, quite a large or lengthy technique is there, and from that quite large and lengthy technique, simply within one page you have to write the food preservation by irradiation. If they are asked for short note 4 marks, 4 marks ला जर short note विचारली तर तुम्हाला लिहिता आली पाहिजे. Yes. Anybody from the class, do you have any problem in the food preservation technique? येथील का लिहिताला जर एसे टाइप प्रश्न विचारला गेला 10 मार्क्सला 8 मार्क्सला, तर थोडक्यात 4-5 मेथड्स मेन्शन करावे लागतील, आणि एक मेथड विचारली तर शॉर्ट नोट्स लिहावी लागेल. येईल का लिहिता बाळांनो? येस
येस सर. ते बीटा रेसचं परत सांगता का सर?
बीटा रेस परत सांगू का? Beta rays may be defined stream of electrons emitted from radioactive substances. They possesses poor penetration power. Cathode rays are an examples of a beta rays, and cathode rays are the same, except that they are emitted from a cathode of evacuated tube used in certain constituents of a food. That is a beta rays. Cathode rays is an example of a beta rays that is obtained from evacuated cathode tube. तर मला बेसिकली काय सांगायचं आहे, अल्ट्राव्हायलेट रेज, बीटा रेज, कॅथोड रेज याच्यापैकी युव्ही रेज किंवा कोल्ड स्टरलायझेशन ह्या दोन टेक्निक बेसिकली लार्ज स्केल वरती वापरल्या जात असतात. UV rays and cold sterilization, that is non-ionizing radiations, are used on a large scale for preservation. तर बेसिकली त्यासाठी तुम्हाला माहिती पाहिजे, काय माहिती पाहिजे की तो कुठल्या प्रकारचे रेज वापरले जातात, त्याची वेव्हलेंथ काय आहे, आणि कुठल्या वेव्हलेंथला मॅक्सिमम इनिबिशन होत असतं ते माहिती पाहिजे बाळा. Yes, anybody is still having any problem? Otherwise, would like to stop over here. Yes. Yes. आहे का काय प्रॉब्लेम बाळांनो? Yes. Is there any problem? Otherwise, next lecture is going to start immediately. Shall I stop over here? Yes.
Yes, sir.
Okay, we'll stop here.